So, we have already seen that the auxin can bring about the cell wall extensibility. It can increase the size of the cell, the elongation of the cell can take place, which can end up in the shoot length. Now, how can we just assess the growth rate? And here you can see a simple equation is being displayed. Growth rate is equal to m into p minus y. Here m is equal to is the irreversible extensibility, p is the turgor pressure and y is the yield threshold. So, based on this calculation you can come to assess the growth rate what is happening in the case of auxin action. When you supply or apply auxin, what level of growth or what is the growth rate which is taking place in the shoot can be assessed from this simple equation. Now, you have already seen that auxin induced proton extrusion, which is acidifying the cell wall, and that is basically helping in the increasing the extensibility of the cell wall, and this is being termed as acid growth hypothesis. And specific proteins mediate the acid induced loosening of cell wall, which we have already seen that it has expansins. Also, hydrolytic enzymes are also activated at this low pH. So, hydrolytic enzymes are necessary for the breakage of the cell wall linkage. So, you know that cell wall is a complex carbohydrate being linked up together, and in order to break that linkage, the action of the hydrolytic enzymes are required, and that is also activated by the action of auxin. So, expansins can cause cell wall loosening in response to an acidic pH. Together with that, the hydrolytic enzymes are acting. Both this can facilitate in the cell wall loosening, which will culminate in the synthesis or in the cell elongation of the cell. So, auxin maintains the capacity for acid induced loosening of the cell wall by uptake and generation of osmotic solutes. So, for the cell wall expansion to take place, not only the hydrolytic enzyme action is enough, not only the expansins action is enough, but the osmoticum has to get built up in the cell, because then only you can see that there is a pressure on the cell wall. When there is pressure, the extension of the cell will be facilitated by the action of expansins and hydrolytic enzymes, which we have already seen, which is getting activated by the action of auxin. And how the osmotic solutes gets built up? You can see that lot of osmotic solutes rush into the cell, or whatever conversion of already stored food material takes place, and these components will add to the osmoticum of the cell, and as a result of that, when osmoticum increases, more of water is taken into the cell, and when more of water enters into the cell, the osmoticum of the cell increases and exerts a pressure on the cell wall, which will finally culminate in the elongation of the cell wall. And you can see that activation of enzymes involved in cell wall polysaccharide biosynthesis is also necessary, because once the cell wall enlarges, the new cell wall materials has to be synthesized and get deposited on the cell wall region. So, in effect, 4 processes are one by one taking place, or simultaneously, I cannot say it is one by one, it is simultaneously taking place. One is the action of the expansins, other is the action of the hydrolytic enzymes. Both will facilitate in the breakage of the cell wall linkage or the components of the cell wall linkage. Together with that, the osmoticum of the cell is being enhanced by the concentration of more osmotic solutes within the cell, and because of that, there is a pressure exerted on the cell wall, which will ensure that the cell wall is set apart, and the cell wall is facilitated by the expansins and hydrolytic enzymes. And that is not enough, the new cell wall materials has to be synthesized, which will get deposited on the newly expanded cell wall region, which will make the cell wall expansion complete. So, these 4 processes are taking place simultaneously by the action of auxin. Again, I will repeat, one is the action of the expansins, second is the action of the hydrolytic enzymes, which will ensure that the cell wall linkages, the components of cell wall linkages are being broken. Third is the osmoticum is being built up with osmotic solutes within the cell. And fourth is the new cell wall materials being synthesized and getting deposited on the cell wall. Now we move into another two important physiological effects of auxins, that is phototropism and gravitropism. We will deal one by one. First we will be seeing the phototropism. Already we started the study on auxins based on the bending of the coleoptile towards the light, and there that is actually the phototropism. The bending of the shoot or the coleoptile towards the light is basically the phototropism. I would like to describe more on this from these simple experiments what you are seeing over here. Here you can see that Chladni Wendt, they are the people who worked on this aspect more, and we call it as Chladni Wendt model. What they have done is that here they have kept an undivided agar block at the bottom, and on the top of it they have placed a cone coleoptile tip, and this is in the dark. And you can see that with the time whatever in the tip had percolated down into the agar block, and in the next case you can see unilateral light was imparted from one end, and again the same happened. Whatever was there in the tip percolated down into the agar block, and if you see almost it was the same. Now in another set of experiments what they did was that they divided the agar block into two. See, they are completely divided with a piece of mica. So here you can see partially the whatever percolated down was almost half, 11.5 and 11.2. So whatever there was there in this particular region came down over here, and whatever over here it has come down here. And here they have not separated it fully. The mica was kept in such a way that it partially enclosed this region, and this at the tip of this region it was open. So once this region is open, there are always chances for the material what is present over here can get transferred into the opposite region, or whatever is over here can get migrate into this region. And accordingly you see that when light is falling, whatever material was there, it gets transferred. Basically now we know it is auxin. The auxin gets transferred into the opposite region of the light which is getting fallen, and accordingly you can see the more of auxin concentration at the bottom. So this was possible since there was no total coverage of this region. So that means the outcome of this studies clearly shows that the auxin is migrating to the tip or the opposite side of the region where the light is falling. If light is falling towards this side, the auxin migrates to the opposite side. And accordingly you can see that here there are some experiments which clearly tell you that here the phototropism is mediated by lateral distribution of auxin. So this experiment had made it very clear that phototropism is the result by the lateral distribution of the auxin. And Chladni Wendt model has clearly shown that the production of auxin is taking place, the perception of a unilateral light stimulus is happening, the lateral transport of IAA in response to the phototropic stimulus is happening. So these are some of the outcomes of the study carried out by the Chladni and Wendt. One is the production of auxin is happening, the perception of a unilateral light stimulus is happening, and the lateral transport of IAA in response to the phototropic stimulus is occurring. And down here you can see that the phototropins 1 and 2, these are basically flavoproteins. These are the photoreceptors for the blue light signaling pathway. So here in the second, again I am going back into the major conclusions of Chladni Wendt, that is the perception of a unilateral light stimulus. There should be something to perceive the unilateral light which is falling to one side of the coleoptile tip, and specifically that is nothing but they are the phototropins, which are flavoproteins. There are two types of phototropins, 1 and 2, and this act as the photoreceptors for the blue light signaling pathway. Phototropin 1 displays a lateral gradient in phosphorylation during exposure to low fluence of unilateral blue light. So, when blue light falls onto that particular region, say for example it is towards this side, whatever flavoprotein is there or whatever phototropins are there get phosphorylated towards this side, and based on that only the auxin is moving towards the opposite side. According to the current hypothesis, the gradient in phosphorylation induced the movement of auxin to the shaded side. So, the gradients of the phosphorylation which is taking place to the phototropins will ensure that auxin is moving towards the opposite side or the shaded side of the coleoptile. Once the auxin reaches the shaded side of the tip, it is transported basipetally to the elongation zone where it stimulates cell elongation and bring about the elongation phenomena which we already described earlier. The acceleration of growth on the shaded side and the slowing of growth on the illuminated side give rise to the curvature, and that is how the curvature is taking place. The more of auxin migrating towards the opposite side, that is the shaded side, will slow down the growth, because I told you earlier the auxin, if it is either exceeding or lowering than a particular optimal concentration, we have seen that the optimal concentration falls between 10 to the power of minus 6 and 10 to the power of minus 5. So, anything exceeding or anything below that can inhibit the auxin action. So, that is specifically what is happening here. More of auxin getting migrated to the opposite side will bring about the slowing of growth on the illuminated side, the acceleration of growth on the shaded side and the slowing of growth on the illuminated side will take place. As a result of that, curvature of the coleoptile tips takes place. Now, coming to the case of gravitropism. This also involves a lateral redistribution of auxin, just like in the case of phototropism. What we are saying in the case of phototropism, there is a redistribution of auxin. From the lighted side, auxin moves to the shaded side. And when there is more of auxin in that region, there will be growth over that region, which will ensure that more it will bend towards the light. So, that is what is happening in the case of phototropism. Coming to the case of gravitropism, here basically statoliths are serving as gravity sensors in shoots and roots. I will go directly to the picture that will be more descriptive. Here you can see this is the normal vertical orientation of the leaf, the root. The root is growing down like this, and based on that you can see the endoplasmic reticulum is lying at the beneath of the cell, and whatever statolith is over the endoplasmic reticulum, what you see over is in light brown colors are the statoliths. So, they exert a uniform pressure on the endoplasmic reticulum. So, the root is growing down just like the taproot system. Based on that, there is a uniform pressure of the statolith on the endoplasmic reticulum. So, that will ensure that there is equal auxin on both sides of the root. Now, imagine a situation when the root, instead of growing vertical, the root is kept in a horizontal position, and that is what being shown here. This is a horizontal position of the root. Once the horizontal position is attained, there is no more pressure of the statolith on the endoplasmic reticulum. See here, there is no more pressure. The pressure is down here. The arrow shows what is the pressure of the statoliths. So, there is unequal pressure of endoplasmic reticulum. Because of that you can see there is more. This is a vertical orientation of the root. Based on the vertical orientation, there is the statolith is placed over the endoplasmic reticulum, uniform pressure, equal amount of IAA towards both sides. So, IAA getting transported, long distance transport comes through the phloem, and based on the pressure of the statolith on the endoplasmic reticulum, you can see equal amount of IAA moves to the both side of the root. But in the horizontal orientation, you can see that the statolith falls down, no more pressure on the endoplasmic reticulum. So, this results in the sensing. Here no light is involved, only the gravity phenomena is being involved, and based on the gravity phenomena, the statolith gets displaced, and based on the statolith displacement and based on the easiness of the pressure on the endoplasmic reticulum, the more of IAA will be transported to the lower portion and less of IAA towards the lower portion. Here, based on the decreased auxin concentration on the upper side, it stimulates the upper side to grow. See here, when there is more. So, what we have seen in the shoot is different in the case of root. In the case of shoot, we have seen the more of IAA getting transported into the shaded side, the more of growth takes place over there, and the shoot bends towards the light side. Here, when there is decreased auxin concentration on the upper side, it stimulates the upper side to grow. So, less of auxin or decreased quantity of auxin is resulting in more growth in the case of root, and when there is more of auxin towards the lower side, the high concentration of auxin to the lower side of the root inhibits the growth, and as a result you can see the bending of the root takes place, and accordingly the root bends and grows down into the soil. So, gravitropism, phototropism phenomena, one way you can say it behaves in a two different way. Here the more of auxin, in the case of shoot, the more of auxin towards the shaded side is resulting in more growth of the tissue and will culminate in the growth of the shoot towards the light. In the case of root, the less of auxin in the upper portion will ensure that the more growth takes place over there, and accordingly the root bends downwards and grows into the soil. So, these are the two important physiological actions of auxin, which will result in the bending of the shoot towards the light and bending of the root towards the gravity. Now, you may ask me whether the gravity perception without statolith is possible, and it is possible. Later the scientists have found out that there is an alternative mechanism of gravity perception that does not involve statoliths and has been proposed in the case of giant celled freshwater chara. There they have found that auxin redistribution laterally occurs in the root cap. So, basically the root cap is involved in the auxin redistribution. No involvement of statoliths takes place, but only the root cap. In addition to functioning to protect the apical meristem, as the tip penetrates the soil, the root cap is the site of gravity perception. So, not the statolith, but it happens in the case of root cap, but only in very few cases the reports are there, and one such report is in the case of giant cell freshwater chara. A chemical messenger is presumed to be involved in communication between the cap and the elongation zone. So, some sort of messenger, chemical message is passing from the cap region to the elongation zone, and based on that the difference in the growth takes place. And basically cap produces a root growth inhibitor. So, root growth inhibitor is produced within the root cap, and here you can clearly see how the root cap is influencing this particular action. Here you see this is a vertically oriented control root with the cap. So, the vertical orientation happens. Now, removal of the cap from the vertical root slightly stimulates elongation growth. So, elongation is taking place, but no bending is occurring. Removal of half of the cap causes a vertical root to bend towards the side with the remaining half cap. So, when half cap is cut out, the bending occurs where the cap is placed and not to the side where the cap is being removed. And horizontally oriented control root with cap shows normal gravitropic bending. And if it is horizontally placed, this is vertically oriented, and when it is horizontally placed, you can see that the cap will ensure that the root will bend down and moves to the soil as per the gravitropic bending. And the same root, if it is kept horizontally but without cap, it will not sense the gravity but will go on elongating in a horizontal direction. So, these are a simple experiments which will show that the cap is very important in these cases, which is signaling or sensing the gravity, and based on that the bending of the root takes place. Now, this is a list of other important physiological effects of auxin. You can see it can regulate apical dominance, it can promote formation of lateral roots and adventitious roots, it can delay onset of leaf abscission, can regulate floral bud development, promotes fruit development, induces vascular differentiation. Also, we know that there are a lot of synthetic auxin applications. We know that it is beneficial for bringing about rooting in the cuttings. You must be aware of solutions which are available in the market, which they sell as rooting medium, and it is nothing but it is a light dose of auxins which is being commercialized. So, if you want to get a quick rooting for the stem cuttings what you are going to plant, you can just take the cutting, dip it in the solution and put it in the soil, and the roots come out very quickly. Flowering in pineapple is also affected by the auxin application, and it is being practiced in big pineapple orchards. Prevention of leaf and fruit development is also happening, or they could do with the auxin application, because premature falling of fruit is a big issue in the case of big orchards. So, you can prevent the premature falling of the fruit in the case of big orchards. Then induction of parthenocarpy, that is also happening with the case of auxin application. And basically people are experimenting, or people are commercially using 2,4-D and dicamba as synthetic auxins for these purposes. Now, this is a very important topic, or we are coming to the end of the auxins chapter, where we will be explaining more on the molecular mechanism of auxin action. And whenever the auxin is being applied, you know that it will come and bind to an receptor protein. And these receptors are mainly localized in 3 regions of a cell, that is on the endoplasmic reticulum, on the plasma membrane, and on the tonoplast. Auxin binding to the auxin binding protein 1, that is a very common auxin receptor which is being localized on endoplasmic reticulum. It is a 22 kilodalton protein, and it is a dimer form. And when the auxin bind to auxin binding protein, it increases the membrane voltage. So, one of the first action of auxin binding is that you can see there is an increase in the membrane voltage. And even when you apply antibody, you can see that the same antibody can come and bind to the this receptor and can prevent the ATPase, it can block the ATPase and result in the hyperpolarization response of auxin. So, if you want to check how exactly auxin is being acting upon, you can use an antibody. Instead of the auxin, antibody comes and binds to the receptor, and it can bring about the same effect what is happening when an auxin comes and bind. Some antibodies, for example, antibody for ABP1 can mimic the effect of auxin by bringing about hyperpolarization of tobacco mesophyll cell, mesophyll protoplast, and stimulation of proton extrusion, etc. So, basically the molecular mechanism of auxin action can be explained based on the binding of the auxin towards the receptor which is localized on the endoplasmic reticulum or on the plasma membrane or on the tonoplast. Now, we will be explaining more regarding the molecular mechanism of auxin action in the next session. So, for the time being, this session I will stop it over here.